Welcome to the PubMed Daily Review for March 23 SD. Today we scanned 997 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5, Surgical Techniques and Patient Outcomes. These papers explore how surgical techniques and patient factors influence outcomes and complications. This cluster covers 173 papers. Representative work includes editorial expression of concern. New technique of end-to-side 2-layered and stent duct to mucosa pancreaticogenostomy with Omental wrapping during Whipple operation. Trunk muscle strength assessment as a predictor of complications in patients undergoing incisional hernia repair, and endoscopic ultrasound-guided treatment of splenic artery pseudoaneurysm and pancreatic pseudocyst. Trend 2 of 5, Biomaterials and Diagnostic Assays. This trend explores novel materials and diagnostic methods for health and environmental applications. This cluster covers 340 papers. Representative work includes efficacy of xanthan-based chlorhexidine gel in peri-implant mucositis treatment, a split-mouth randomized clinical trial, comprehensive characterization of solution cast polycaprolactone/emzine/gelatin composite films for biomedical applications, and assessing the clinical diagnostic utility of multiplex dPCR assays in thyroid nodules. Trend 3 of 5, Neurocognition, Biomarkers, and Health. This trend explores how biological markers and cognitive processes influence health outcomes across the lifespan. This cluster covers 132 papers. Representative work includes mortality predictions, including pre-admission functional status in ICU patients with delirium, a sub-study of the 8-ICU trial, altered resting state functional connectivity in women survivors of intimate partner violence, an ICA study, and the relationship between dietary sodium intake and cognitive function, a narrative review. Trend 4 of 5, Cardiac Health Monitoring and Prediction. These papers explore diverse methods for assessing, predicting, and managing cardiac conditions, from wearable tech to advanced imaging and genetic insights. This cluster covers 78 papers. Representative work includes remote monitoring of heart failure exacerbations using a smartwatch, correction, exercise rehabilitation for heart failure and associated cardiomyopathies, and the comparison of cardiac and body morphometry in patients with non-dipper and reverse dipper hypertension, a retrospective cross-sectional study. Trend 5 of 5, Patient Engagement and Health Technology. These papers explore how technology and patient perspectives intersect in healthcare and health-related behaviors. This cluster covers 274 papers. Representative work includes the disengaged patient, chronic interfacing in a regime of digital health convenience, the folk's sociological imagination, manufacturing agency through smoking among Chinese adolescents, and multidisciplinary Delphi consensus on management of children with moderate-severe atopic dermatitis. Up next, our first paper out of 5, Single-Cell Multiomics uncovers an endothelial mechanosensitive piezo 1L 33-axis driving pulmonary fibrosis by Lanlan Zhang and Shanzichun from Respiratory and Critical Care Medicine, Center for High Altitude Medicine, State Key Laboratory of Biotherapy, West China Hospital of Sichuan University, Chengdu, Sichuan, China. Let's break down its key contributions. Pulmonary fibrosis is a serious lung condition where the lung tissue becomes stiff and scarred. We know that the cells lining our blood vessels, called endothelial cells, are involved in this scarring process. However, we haven't fully understood how physical forces acting on these cells might contribute to the disease. This study aimed to uncover that connection. To investigate this, the researchers used advanced single-cell multiomics to analyze both human lung tissue from patients with fibrosis and experimental models. They specifically looked at the role of a protein called piezo 1, which senses mechanical forces. They found that piezo 1 was more active in endothelial cells in fibrotic lungs. To confirm its importance, they genetically removed piezo 1 from endothelial cells in mice and observed the effects on fibrosis. The key finding was that piezo 1 was significantly upregulated in endothelial cells within fibrotic lung tissue. Crucially, when they removed piezo 1 specifically from these cells in mice, the development of lung fibrosis was significantly reduced. This strongly suggests that piezo 1 activation in endothelial cells is a driving force in pulmonary fibrosis, acting through a specific molecular cascade involving IL-33. This research highlights a previously unrecognized mechanism where mechanical forces on blood vessel cells contribute to lung scarring. The endothelial piezo-1 IL-33 axis emerges as a promising new target for developing treatments for pulmonary fibrosis. Future studies will likely focus on how to safely and effectively modulate this pathway to help patients. Up next, our second paper out of 5, Bimagrumab plus semaglutide alone or in combination for the treatment of obesity. A randomized phase 2 trial by Stephen B. Hamesfield and Kenneth M. Addy from Biomedical Research Center, Louisiana State University, Baton Rouge, Louisiana, USA. Let's break down its key contributions. Obesity is a complex chronic disease affecting millions worldwide. While lifestyle changes are foundational, pharmacological interventions are crucial for many. Bimagrumab, a novel antibody, shows promise in modulating body composition. This study aimed to evaluate its effectiveness, both alone and in combination with semaglutide, a well-established weight loss medication. This was a rigorous double-blind, placebo-controlled phase 2 trial. We enrolled 507 adults with obesity and randomized them into 9 different treatment arms. These included placebo, Bimagrumab at 2 doses, semaglutide at 2 doses, and various combinations of these drugs. The primary goal was to measure the change in body weight after 48 weeks, with an extension to 72 weeks. The results were compelling. The high-dose combination of Bimagrumab and semaglutide achieved an impressive average weight loss of 17.8 kilograms at 48 weeks, significantly outperforming placebo, p less than 0.001. Bimagrumab alone showed a 9.3 kilograms loss, while semaglutide 2.4 milligrams resulted in 14.2 kilograms loss. Importantly, the safety profiles were consistent with what's already known for each drug. This trial demonstrates that combining Bimagrumab with semaglutide can lead to substantial weight reduction in individuals with obesity. This dual-action approach holds significant promise for future obesity treatment strategies. While these findings are exciting, further investigation in larger, diverse populations is warranted to fully understand its long-term impact and optimize its use in clinical practice. Up next, our third paper out of 5, Single-Cell Atlas of the Developing Down Syndrome Brain Cortex by Michael Latka and Vincenzo De Paola from Brain Sciences, Imperial College London, London, UK. Let's break down its key contributions. Down syndrome, caused by an extra copy of chromosome 21, is the most common genetic cause of intellectual disability. While we know it affects brain development, the specific molecular changes that occur during fetal development remain largely unclear. Understanding these early disruptions is crucial for identifying potential therapeutic targets. To investigate the molecular underpinnings of Down syndrome brain development, researchers analyzed approximately 250,000 cells from fetal cortices of both Down syndrome and control individuals. They employed advanced single-cell, transcriptomic, and chromatin accessibility profiling techniques. This allowed them to identify specific cell types affected and map the gene regulatory networks involved. The analysis revealed a significant subtype-specific reduction in excitatory neurons expressing RORB and FOXB1. Furthermore, widespread disruption of normal neurodevelopmental gene expression programs was observed. Key transcription factors on chromosome 21, including BOC1, PKNOX1, and GABA, were identified as critical dosage-sensitive regulators. Importantly, experimental manipulation using antisense oligonucleotides showed a partial rescue of target gene expression, suggesting a potential therapeutic avenue. This study provides an invaluable resource for understanding how Down syndrome impacts brain development at the cellular and molecular level. It pinpoints specific genes and pathways that are disrupted early on, offering crucial insights into the mechanisms underlying intellectual disability. The findings open exciting possibilities for developing targeted therapies aimed at normalizing gene expression and improving neurodevelopmental outcomes in individuals with Down syndrome. Up next, our fourth paper out of 5, Major Adverse Limb Events and Death After Successful Endovascular Revascularization. Best CLI trial by Scott Kinley and Richard J. Powell from Interventional Endovascular Therapy, Cardiovascular Division, Department of Medicine, VA Boston Healthcare System, Boston, Massachusetts. Let's break down its key contributions. Chronic limb-threatening ischemia, or CLTI, is a serious condition that can lead to amputation and even death. Revascularization aims to save limbs and improve quality of life. While endovascular approaches are common, we need to better understand what factors might lead to adverse outcomes, even when the procedure itself is successful. This study looked at patients from the BEST CLI trial who had a successful endovascular procedure for CLTI. They analyzed data from 773 patients, examining their baseline characteristics, the details of their leg lesions, and the specific endovascular techniques employed. The researchers followed these patients for an average of 2.7 years to see who experienced major adverse limb events or death. The analysis revealed several key factors associated with major adverse limb events or death. Patients with end-stage renal disease faced a significantly higher risk, as did those with wounds located at or above the ankle. Longer procedure times were also a predictor of worse outcomes. Interestingly, the use of advanced endovascular technologies like drug-coated balloons or stents did not appear to be consistently linked to these adverse events. This study highlights that patient-specific factors, such as kidney disease and the severity of limb wounds, are strong indicators of potential complications after endovascular procedures for CLTI. The findings suggest that various endovascular techniques, even advanced ones, can be safely employed when needed. This underscores the importance of thoroughly assessing patient risk factors to guide treatment decisions and optimize limb salvage strategies. Up next, our fifth paper out of 5, cross-kingdom mRNA delivery by Panax notoginseng-derived extracellular-like nanoparticles vesicles restores neuronal function after ischemic injury by Yuan Yu and Hongxing Mai for neurology, Sun Yat-sen Memorial Hospital, Sun Yat-sen University, Guangzhou, 510120 China. Let's break down its key contributions. Ischemic stroke is a devastating condition, and getting treatments across the blood-brain barrier is a major hurdle. But what if we could use nature's own delivery systems? This research explores plant-derived nanoparticles, specifically from Panax notoginseng, as a novel way to deliver therapeutic agents directly to the brain. The researchers isolated nanoparticle vesicles from several medicinal plants, calling them PEVs. They found that vesicles from Panax notoginseng, or NOTO-EV, showed the most promise. Using both cell cultures and animal models of stroke, they investigated how NOTO-EV worked. They discovered that these plant vesicles delivered microRNAs into neurons, influencing key cellular pathways involved in stress and survival. The results were quite impressive. NOTO-EV treatment led to a significant reduction in the area of brain damage, preserved the structure of neurons, and importantly, restored electrical activity. This was achieved by the plant nanoparticles delivering their cargo, which then activated crucial cellular pathways like mTOR and helped stabilize mitochondria, ultimately protecting the neurons. This study highlights a groundbreaking approach to stroke treatment using plant-based nanotherapeutics. The ability of these NOTO-EVs to cross the blood-brain barrier and deliver functional microRNAs is a significant advancement. While promising, further studies are essential to translate these findings into a viable clinical therapy for patients suffering from ischemic stroke.